So hello everyone, and welcome back to part number two of chapter four. So in the first part, we talked a lot about different types, of organelles. So we started off with basically cells, to the cell theory. So what the cell theory consists of, and then about microscopes, especially how we use microscopes to study cells, and how does the microscope actually allow us to determine the structure and a lot about the function of, the cell by itself, but other types of organelles. We spoke about prokaryotes quite well in depth, and we then spoke about partly about eukaryotes. So now in part two, we're going to continue on to fully describe eukaryotes. So as I was said, the biggest difference with eukaryotes and prokaryotes, are multicellular. They actually have a nucleus, while prokaryotes have a nucleoid, so different structures. And eukaryotes have all these different types of organelles. So these organelles, which are basically, have this kind of membrane that helps in the compartmentalization in the cell, along with the endomembrane system that helps compartmentalizing um, basically functions in the cell. So we'll continue on and we'll start off back with mitochondria. So mitochondria, as you guys have most probably heard, in high school, it's the powerhouse of the cell, right? It's the organelle that has a big function in giving us energy. And they have a very weird structure, very unique and particular structure, and where they actually have two phospholipid bimembranes. The first one, which is shown here, is this outer membrane, which creates this really nice, smooth, kind of capsule-like structure. Then you have the inner membrane, which is shown basically here with my cursor, and this one is a bit more folded. It actually is quite folded compared to the outer membrane, and these folds are called crista. So the folds are the crista, as you saw as shown here. So this is basically an electron microscope. As you clearly see here, the outside is this really nice smooth membrane, and then you have these kind of folds, these crista all around, and that's the second inner membrane that exists in the mitochondria. So basically, what happens because of these two membranes, you have two empty spaces. The empty space that is between the inner and the outer membrane, which we call the intermembrane space. So it's the membrane in between the inner and the outer membranes. And then you have another empty space, which is inside of the inner membrane, which we call the matrix. So these are just remember these locations because hopefully when we start going to chapter 7 and 8, when we start talking about how we create ATP in the mitochondria, basically where these reactions start to occur, it will be in certain locations. So it's good to know just basically what we mean by the matrix or the cresta or the intermembrane. So keep that in mind. But mitochondria is quite particular that it actually has its own ribosomes stuck to each other. But you can clearly see here these kind of little O's, like small little circular objects. That is actually DNA. So mitochondria actually has DNA inside of it. So DNA is not only found in the nucleus, but it's also can be found in the mitochondria. And a lot of this DNA actually encodes for proteins that carry out oxidative metabolism. So this all is metabolism that helps us actually create energy. So it's quite interesting and unique. However, of course, throughout evolution, the mitochondrial DNA actually, it has it encodes for all these proteins for oxidative metabolism, but to carry its full function, it still needs nuclear DNA. So right now, if I actually take the mitochondria by itself and I put it, in a test tube, it's not going to grow by itself and it's not going to carry out its full function because it still needs proteins that are encoded from the nuclear DNA. So it's not, autonomous. That's the thing that I'm trying to get into. So it has its own DNA, but it's not fully autonomous. It can create certain proteins to do certain functions, but not at its full capacity. So one thing about this mitochondria is that during division, of course, you need to have two of them, right? So one goes to one cell and the other one goes to the other during mitosis. So what it does, it actually just increases its membrane into like a bigger structure, and then it cuts in half. So you get two of them. So that's how one way of basically, they divide. So one other very important organelle that exists in eukaryotes is chloroplasts. Chloroplasts are basically found. mostly are just found in plants and some other eukaryotes, but mostly in plants. And these guys are much bigger than the mitochondria, actually more complex than the mitochondria. These guys are very involved in transforming light into energy. So they're bigger than the mitochondria, but they have very similar kind of structures in that they are actually made of two membranes, again, an outer membrane and an inner membrane, as shown basically here. And then when we go and look inside the the chloroplast, you have these massive stacks, which we call granum. And these stacks are made of discs, as you see here. So, for example, this is made of two stacks, and this stack is made of two discs. This stack is made of three discs, and the disc we call them thylakoid, thylakoid discs. That's what we call them, and basically they're covered in a membrane that we call also thylakoid membrane. And these, so the thylakoid discs pair on top of each other to create a granum, and these are the ones responsible in actually carrying out all their cellular reactions that converts uh, light into energy. So keep that in mind. Also, one thing that is very similar with chloroplasts and mitochondria is that they also have their own DNA. So they can actually have their own genetic information built in there. But then this brings you into some theory. Why does the mitochondria, the nucleus, the chloroplasts have their own DNA? Isn't that a bit weird? Shouldn't there, like, just be one place where we store all the DNA and that's it for one cell? Just the nucleus? Why does the mitochondria have DNA? Why does the chloroplast have it? And this made us really think as scientists to start like, okay, there's something peculiar here, and most probably something has happened in the past that gave this, gave mitochondria DNA or chloroplast DNA. What is this? And they came up with this endosymbiosis theory, meaning that most probably what happened was, kind of old-fashioned cells. So these are, let's say, primitive cells, so most probably prokaryote cells, kind of came together and kind of join forces together to create, let's say, the modern cell. So the modern cell of the eukaryotes that right now, these advanced cells that we have in eukaryotes, most probably comes from two primitive cells, most probably prokaryotes, that came together and fused to create this one more advanced and technical cell that we call the eukaryote cell right now. So this came as the endosymbiosis theory. So it proposes that some present-day eukaryotic organelles evolved by a symbiosis between two free-living cells, meaning that an eukaryotic cell that has these very advanced organelles, it's most probably because of two free-living cells that came together and they basically join these organelles together. So you have one big cell that has these advanced organelles together. So, what it basically means is that, you have a cell that basically that engulfs another cell to, give, more organelles to one cell. So that's what basically we're trying to say. So there is basically one cell that's engulfing another to kind of, incorporate this newly added organelle. So there are two possibilities of what is the engulfing cell. So what is the first cell that first engulfs the other cell to create basically, the modern version of a eukaryote. The first theory or possibility is that the engulfing cell is actually an archaean. So it's an archaean, so a prokaryote that engulfed some other, prokaryote that gave basically gave mitochondria or chloroplast. That's what we thought. Or the second is that no, is that actually a bacteria first engulfed an archaea, which gave up the nucleus. So then this cell, that it was basically bacteria and archaea, engulfed another cell that basically gave either the mitochondria or the chloroplast. So that's where it came from. So if you think about this, like what made us come to these conclusions is that okay, so mitochondria and chloroplast have their own DNA. Bit weird. So these things like most probably some sort of they were previously a different cell, but then we started to realize what really gave us a lot of evidence for this theory was that there were a lot of prokaryotes that actually had exactly the same functions of just the mitochondria or chloroplasts. So it's like most probably that's what it is. That these guys, that these prokaryotes that only had these kind of functions of a mitochondria or chloroplast were most probably engulfed to create this bigger and advanced cell. what was that engulfing cell is, these possibilities that I was speaking about. So now let's move on to cytoskeleton. So if we take right now a cell and we cut it in half, if you cut it in half and you do some really nice electron imaging, what you're going to see is a track. You're going to see this crisscrosses of these big, massive kind of filaments going all over the cell. And these, if you want to compare them to something, they actually look like roads and bridges and all this infrastructure where we basically use our cars to actually, drive in. So that's how they look like. And this is what they actually became to be. So these are proteins, these big filaments of proteins that is in the cell that of course give structure, but they also lay a foundation for cargo in the cell to move around, but also to help the cell move. And there are basically three types of cytoskeletons in the cell. There is microfilaments, microtubules, and intermediate filaments. So let me start first with microfilaments. Microfilaments are the smallest actually filament. They are 7 nanometers in diameter. So this is how big in the diameter they are. And they're composed of these actin proteins. So actin is a very globular protein, as I'm showing here with my cursor. It's this really big ball. And what happens is that they actually, polymerize with each other. So they become a polymer. So polymer is literally, again, a big structure that are made of simple monomers. So that's what they are, is monomers of, actin all joined up together. So you create one filament. And what happens is this one filament then combines with another filament to create, a structure made of two actin filaments shown here. So this actually looks like kind of a pearl necklace that you see on your grandma or whatnot. So they're quite interesting structures, and these are super important actually for cell movement, specifically contraction and crawling. So if you see kind of cells that are like crawling, this mostly because of the actin. And these also help in pinching. This is a process you'll definitely start to learn more in mitosis. So when the cells start to divide and they want to divide into two cells, there is this membrane that pinches and kind of cuts off. And it's all because of actually the microfilaments. These actin filaments are basically, cutting it in half and kind of giving this pinching kind of like motion. So that's what they're important. Now we have a second type, which are microtubules. Microtubules are much bigger than microfilaments. If I'm not mistaken, they are around, so let me go quickly here to your textbook just to make sure that I don't say something that's wrong. So it's 25 in nanometers in diameter. So they're much bigger actually than the microfilaments, the microtubules. And they're basically what they're made up of these big hollow tubes, which are 25 nanometers in diameter, and they're made of 13 filaments. Each filament is actually made of dimers of alpha and beta tubulin. So as you can see here, which are shown in different shades of green, they're basically, alpha beta subunits creating one big filament, and this filament, 13 of them are creating this hollow tube of 25 nanometers of diameter. So these are microtubules. These guys, literally, you can think about them as kind of bridges. They're literally these bridges that help cargo be transported from one place to the other. That's their biggest function. And then you have intermediate filaments. Intermediate filaments are a bit more tougher than microtubules or microfilaments. These are fibrous proteins like keratin or vimentin. So let me show you if we can see other examples. They're keratin, vimentin, and other types of proteins that we basically have in our cell that help and give structure. And these guys are usually not broken down because we don't want them to break down. All we want them is to be stay there and help us, give structure to the cell. Microtubules and microfilaments are actually they break down much easier. Actually, microtubules are broken down quite quickly. If you have a dividing cell, they break down every 2 minutes or so. If not, they break down every 20 minutes or every 20 minutes. So they're not, stable like intermediate filaments. And same thing goes with microfilaments. They keep breaking down and forming quite constantly. While intermediate filaments, not really. They're usually very stable and not broken down. And intermediate filaments are bigger than microfilaments. I think they're between 8 and 15, if I'm not mistaken. sorry, they're 8 and 10 nanometers actually in diameter. So you have microtubules being the biggest, then come your intermediate filaments, hence their name, intermediate, so in the middle, and then micro, hence comes the micro, very small filaments. So that's basically what your cytoskeleton is made of these three different types of filaments. So now we have another actually kind of, let's say, organelle, which we kind of like to call the centrosomes. The centrosomes are kind of made of basically these two centrioles, which are basically these two structures, and they're basically exactly the same. They're two centrioles, and they actually are facing each other at a almost 90-degree angle. So this kind of right angle. So this is literally how they are kind of positioned in that kind of sense. And they are actually made of microfilaments. And in specific, again, there are this hollow-like structure, and they're made of basically triplets of microfilaments, and they're arranged in nine. So you have three basically microtubules that are basically joining together. So sorry, basically centrioles or centriole is made of microtubules, not microfilaments, microtubules. So there are three microtubules creating one structure, and there's nine of them creating this hollow tube. So as you can see here, 1, 2, 3, 4, 5, 6, 7, 8, and 9. And you create these nice hollow tubes. But around these basically, so this is basically how they look like around here, they have this membrane, which we like to call this peri pericentral central material. So there's this basically material all around these two centrioles. This material is technically alpha and beta tubulin. So we're like, okay, so these guys are microtubules. They're surrounded by the building blocks of microtubules. Hmm, these most probably are the organizing centers. So basically, these guys, their function is to create these big microtubules, and that's what they are. They're the center, they're the nucleating center of these microtubules. So these guys will basically start taking alpha beta tubulin from the pericentricular material and start creating these big filaments. So usually when you start creating these filaments, you have polarities. You have a plus and a negative end. So polarity, these ends, they're not meaning they're negative or positive charge. No. Negative, what it means is that it's closer to the central, to basically the nucleating area. Positive is growing outside. So this is the filament that's going outside. This is the part that's mostly more further away than the centrioles. So negative is basically the side that's closer to the nucleating area, so the centriole, and the positive is the one, the basically the side that's going outside of it. So keep that in mind. Positive out, negative more in. So that's how to keep that uh, basically memorized. So I was saying is that these microtubules, they are literally bridges. Because what happens, as we saw in the endomembrane system, you have a lot of cargo, right? And this cargo is going from, it's actually going from the nucleus to your ER, from your ER to your Golgi apparatus, and from your Golgi apparatus to the plasma membrane. So these vesicles need to move, right? They need to move either actually, outside to the plasma membrane, or even sometimes back to the Golgi or back to the ER. So you need this network to do so. So you basically start to build these massive bridges that we like to call microtubules. So there's big, nice structures that are made of 13 basically filaments, as we were talking about microtubules. And of course, this helps to create a foundation to transport the vesicle. But of course, the vesicle by itself cannot just move into the microtubule. It needs another protein for that. So you need a car. So think about this is that the vesicle is one big container that you cannot move. It's too big. So you need a car. And this is exactly what these molecular motors are. An example of the molecular motor is dynein. So you see this dynein over here. This is the car. And this car actually has a dynein actin complex. So this is a protein that connects to dynein and the vesicle. So its main function is simply to connect to dynein and the vesicle. So now these are connected to each other, and dynein is then able to move along these microtubules thanks to ATP. ATP gives it energy to move along. Dynein actually will move towards the minus end. So these move towards the nucleating areas. So these move towards the nucleus. So if you need vesicles that are, let's say, going from the Golgi and you want them to go to the nucleus, you use dynein. But if you want them to move towards the plasma membrane, for example, from the Golgi to the plasma membrane, you need a different type of motor that goes to the plus side, so towards more the plasma membrane. So in this case, you need, these guys are called actually kinesins. I'm gonna find them here. Yes, they're called kinesins. So kinesins go towards the plus, while dynesins move to the opposite direction. So basically, as you can see here, dynein moves towards the minus end, so inward toward the cell's center, so the nucleus. While kinesins the opposite. They go more to the cell periphery. So they're basically going to the plus end. So that's how things are able to move back and forth because of these microtubules and also the molecular motors. So one thing, of course, is that is extremely important for a cell is to move, right? And the greatest example for this is your immune system. Your immune system needs to sometimes move around to all parts of the body because let's imagine, your immune system is sitting down, let's imagine in your thymus. So this is a small organ that is actually found near your neck. So sometimes what you need is that the cells that are sitting there to go down to the toe of your foot because you have an infection. Bacteria are there, so they need to move. How they move is through crawling. So this movement of crawling is literally like how you see a baby crawl when they're putting literally their arms forward and kind of pushing themselves forward. That's exactly how a cell moves. And you actually can see these actually here on the YouTube um, kind of links that are here, and you clearly can see these cells actually moving like that. And this is all thanks to actin. So what actin does, it will start to polymerize right next to the cell membrane. So it's going to push the cell membrane forward, literally like extending the arm of the cell membrane to go forward, like a baby extends its arm forward. And then you have proteins that are contracting on the actin filaments to push, all of the cell forward, just like the baby does. It puts its arm forward and kind of pushes itself forward. Same thing goes with these cells. So actin filaments kind of push the cell membrane forward like an arm, and then you have these proteins that are basically kind of contracting the cell to move forward. That is quite beautiful to see, actually. And this is how cells can move. Another one is through flagella. So remember these flagella, these big structures that we were talking about in prokaryotes. These like very big structures that are help propel cells forward. Well, eukaryotes also have flagella, but these guys are much different than the prokaryotes in how they're actually composed of, their structure, and whatnot. So as we saw before, you clearly saw in the prokaryotes that the flagella is actually extending outside of the cell, while when it comes to eukaryotes, this flagella is actually a projection of the plasma membrane. So you have, first of all, this basal body, which is actually the cytoplasm, and then it starts to create this big filament that we call the flagellum. And this one is actually pushes through the plasma membrane, so it's a projection of the plasma membrane. So that's why it's actually wrapped in this plasma membrane, this whole flagellum. So you have the basal body shown here, and it has this following structure. It actually is again made of microtubule, this triplet microtubules, and there's basically nine of them. Hmm. Triplets making nine kind of structures to create this big tube. Where have we seen this? Well, it's basically very similar to the centriole structure, and that's what actually it does. It's similar to the centrioles that they create or nucleate, or these big filaments. That's what it's doing. It's creating the flagellum, and the flagellum is literally microfilaments that are extending to create the flagellum. And it gives this structure. You have doublets, not triplets. In this case, you have doublets that are organized in nine. So you have doublets that are, there's nine of them to create this big structure in the middle. And inside, you have a doublet. So that's why we call this actually this 9 plus 2 structure. So nine outside. So you have nine doublet microtubules, and then you have two in the middle. So you have just a doublet in the middle. That's the flagellum. And one thing that's a bit different actually in the motion So prokaryotes, as we saw, they rotate, clockwise, while this guy over here rotates actually up and down. So it's not different movements. This one goes, it kind of folds around up and down, very different than rotating. So that's one thing. However, eukaryotic flagellum actually, a lot of eukaryotes have actually lost them. We don't, for example, in animal cells, we don't have this flagellum. We see this very similar structure actually in cilia. So cilia are these small hair-like projections that you see in your throat or actually in your digestive tract. And these what these guys do, they flop back like up and down. So very similar to basically the kind of the movement of the flagellum. And they basically what they do, these guys, is that they kind of try to remove mucus or any bacteria from these areas. And they have exactly the same structure, this 9+2 structure. So there's where we can see the structures actually in humans in the cilia. Flagellum, we don't have it in animals. So just keep that in mind. So then you have eukaryotic cell walls. So one thing that was very unique for bacteria is that they have cell walls. Well, in eukaryotes, you also have cell walls. In plants and fungi and some protists, we also have cell walls, but they're chemically and structurally very different. So and protist cell walls are made of cellulose, while fungi cells are actually made of chitin. Bacteria cell walls are actually made of a different subunit, which is basically this N-acetylglucosamine. So it's a different type of glucose monomer that gives a different structure. While eukaryotic cells are, made either, plantoprotists or cellulose, fungi is more of chitin. But its structure is different. So let's take a look, for example, the plant cell wall. You have basically this is the plasma membrane, as you can see here, and I'm showing with the cursor. And then you have this primary cell wall. But in between the primary cell wall and the plasma membrane, you have a secondary cell wall shown here also. So that sometimes they're made of two cell walls actually. And then here, as you can clearly see, within this is one plant cell. You have another plant cell. You have these small corners, which are actually made of a substance called middle lamella. This is this very gluey substance that helps to actually push or kind of glue cells together. So that's how the structure of the cell walls are. So you have these two cell walls, and they're actually glued to other cell walls because of this middle lamella. So that's how the cell walls are actually, as you can see in the eukaryotes, a bit different to bacteria. In animal cells, we don't have cell walls. We have something called extracellular matrix, in where what we do is we like to actually release a lot of glycoproteins to the space outside. So this is the plasma membrane. What we do is release a lot of these kind of carbohydrates, these glycoproteins like collagen, elastin, proteoglycans, all these big, massive macromolecules that kind of help the cell have support, strength, and resilience. So you have the cell plasma membrane, and then you have another zone that's not connected to the cell membrane. So it's not connected. It's just a bunch of proteins and glycoproteins that are kind of giving this nice protection. And this is very important actually, because when you start learning about microbiology and in specific virology, viruses need to secrete actually enzymes that are able to cut through this extracellular matrix to make it to the cell. So that's one thing to keep in mind. So now, thankfully, with the chapter part one and part two, you guys are able to actually fill this whole table. So, in the exam, what I can do is that I can actually put all of these basically, different types of organelles or whatnot, and I can put prokaryote, animal, or plant. And what I'm expecting from you is that you guys will actually know is if it's present or absent. This is what I need you guys to know because this is um, it needs to be known because of course I've spoken about every single one of these guys over here, and I also stated what they are and a bit more of the structure. So that's one thing I want you to learn. Sorry guys, I'm a bit thirsty, that's why I'm having these pauses. Okay, so one last concept that I want to actually go through here, which is cell to cell interactions. So cells have to communicate with each other, right? So you have a tissue, and a tissue is basically, the same cells that are made, that come together to create this tissue that has basically the same function. So cells have the same function, come together to keep a tissue. right? So these cells need to interact with each other. They need to communicate with each other, and they need to sometimes be identified. They're like, okay, I'm this type of cell. You're that type of cell. Pure example in my lab. So I'm giving this example, and please do not memorize the example I'm giving right now because this is a bit too complex. But I want to tell you, how important this is. So in the immune system, you have tens of different types of immune cells. There's so many different types of immune cells. How am I able to actually in immunology to distinguish what type of cell am I dealing with? Well, it's all because of surface proteins. What proteins is there actually in their plasma membrane? That's one thing how I can do it. Pure example, and these are the examples that you do not need to actually memorize, are for example, if I see a cell that has CD3, oh, it's a T cell. CD19, B cell. If it has, CD56, oh, damn, this is actually a NK cell. CD14, this is a monocyte. Oh, this actually has, LY6 G. This is definitely a neutrophil. Oh, this has a gamma delta TCR. Oh, this is a gamma delta T cell. These are the things that I actually then start to look in the cell and tells me the identity. Well, this is you see this actually a lot. So one example is glycolipids. So for example, in red blood cells, why do we actually call some blood cells type O, type A, B, or AB? Is depending on what glycolipid they have in their surface. If they have this one glycolipid, we call this, oh, they're group A. If there's this other one, is type B. If they have it together, we call it AB because of the surface proteins. And one thing that also, the immune system does, as we saw, is one thing it likes to go always scan our body to see, recognize cells that are, self or that are non-self. Because the ones that are self, it doesn't harm them. But the ones that are non-self, so most probably these guys are pathogenic. So these guys, are, they're trying to do something or cause disease to us, they have to recognize it and kill it. So it actually these immune cells bind to MHC cells that are present on these cells, and this MHC it tells it if it's self or non-self. So that's one thing actually how surface proteins are able to interact to identify each other. Well, one thing that you also need sometimes in cells is that you need them to be connected with each other. Sometimes you need them to be very, like let's say connected very tightly or non-tightly, or you want some sort of connection that allows certain ions or certain building blocks to pass through. And there's three ways to do this. There's adhesive junctions, septate or tight junctions, or communicating junctions. I'll first talk about adhesive junctions. Adhesive junctions are proteins that mechanically attach cytoskeletons of neighboring cells or cells to the basically extracellular matrix. And I'll give you an example of this, which is cadherin. Cadherin is one of the most primitive proteins that has existed for decades or millions of years, basically. And what these guys are these big proteins on the plasma membrane that basically are connected to the cell cytoplasm. So they're sorry to the cytoskeleton. So in this case, for example, here it's connected to actin. And what it does, it connects to an adjacent cell cadherin. So you have here, this is one cell and cell two. They're connecting basically the two cadherins to each other. And these cadherins are connected to, this is to actin, and this is basically to the tubulins. So that's how these two cells, cytoskeletons, are connected through these cadherins. And this what helps basically that this actin and this beta microtubules are connected to each other. So the cytoskeletons of these cells are connected. And this is very important in cells where there's a lot of contraction, like the heart. We'll see this a lot in heart. Another one are the septate or tight junctions. So these guys, what they basically do, which are shown here, they help cells connect with each other super tightly, just like its name said. And I'll give you an example to actually, go into depth here. These tight junctions, you clearly see them in the digestive tract of animals. So like us, if you look at our digestive tract, it's actually one cell thin. That's how thin it is. It's just literally one cell. It's made of one cell. It's not made of multiple layers of cell. No, no. It's just one cell. This is a bit dangerous if you think about it because through our digestive tract, there's a lot of food that passes through. And one thing that you do not want to happen is that the food that we want to absorb is passing through cells. You don't want that because if it passes through the cells, we're losing all that kind of important building blocks that we need. So you want to make sure that these cells are super well connected and that there's no actually open space. And this is the thing of tight junctions. These are these proteins that act like a belt. They wrap around these cells and they force them to come together and basically almost kind of prevent any opening to happen. So I'll give you an example of what these proteins are actually. So here they are. You have them, um, these tight junctions. So these are called claudins. Claudins. So these claudins create these big, kind of let's say belt-like structures around these two cells, and they push it to come so tight with each other. And this really helps, for example, in the digestive tract to make sure that building blocks pass through the cell, no, sorry, into the cell and not through it, not through these opening spaces. So that's what tight junctions can be used for. And then you have basically another type, which is the communicating junctions. In communicating junctions, as their name are, there are these kind of junctions that help two cells communicate with each other. And how cells communicate with each other is through signals like, passing of ions or passing of certain building blocks like glucose and whatnot. So you can have these kind of, let's say, kind of pores like this that allow things to pass through. We have this definitely in animal cells and invertebrates and vertebrates, and there are different ones. For example, I'll give you the examples here. In invertebrates, they're formed of proteins that are called pannexins, and in vertebrates, they're called pannexin-based cap junction exist, but they also have these connexons, so mostly made of connexons in vertebrate uh, cells. In plants, it's quite interesting. We call them these plasmodesmata, and these are actually just extensions of cell membranes or just membranes. Could be here, for example, of the smooth ER that's extending and protruding to the other cell, and allows this basically passage of certain, for example, calcium in this case, because smooth ER has a lot of calcium, so allows the calcium go from this to here. And this also only happens to these kind of specialized openings in the cell walls. It's not going to happen randomly. There's specialized openings in cell walls between cell number one, cell number two in plants, and it's literally just these membranes that are crossing from cell one to cell two. So that's how are able to talk. So we now come to the end of this lecture. So we spoke a lot about all the different types of organelles, the cells, different types of cells, prokaryotes and eukaryotes, and how they differ, and all the different types of eukaryotes. Sorry, different types of organelles found in eukaryotes. So please, guys, if there is any question or anything that you guys do not understand, please let me know. I am open to anything. My, let's say, office hours online are unlimited. Please send me whenever you want, and we can always, of course, talk through the email. Or if that is not enough, we can, of course, make a Zoom. Or in cases of there's no internet, proper internet, we can also have a call, and I can actually discuss certain things. So hope this helps you, and hope everyone's safe. Thank you so much for tuning in.